See, today's class, it is a summary of the entire chapter, cell cycle and cell division. Okay. So in this chapter, we studied about the different concepts like what is growth. Growth is an irreversible increase in the size and mass of an organism with reference to time. So how is growth and reproduction brought about? Growth and reproduction of all the organism, they depend on the cell division and cell enlargement. So they depend upon two important aspects, growth and reproduction, cell division and cell enlargement. Okay. So even in the formation of gametes, there has to be cell division. So in the formation of gametes, usually it is the meiotic cell division. And in the growth, it is generally going to be the mitotic cell division. So growth and reproduction of all the organisms depend on the cell division and cell enlargement. So the mechanism of cell division and multiplication of cells together they constitute the cell reproduction. So the mechanism of cell division and multiplication of cells together they are going to constitute the cell reproduction. So that is what you have to remember about. So the uh, mechanism which involves cell division and multiplication of cells together we can define it as uh, cell reproduction. So cell reproduction involves two mechanisms, cell division and multiplication of cells. So that is what is the definition of cell reproduction. So in the defined cell cycle, it is the life period of a cell during which a cell synthesizes DNA by replication. The cell grows and it divides into two daughter cells. So this entire life period of a cycle, we term it as cell cycle. So cell cycle is the life period of a cell during which a cell synthesizes DNA by the replication. It grows, the cell grows, and the cell divides into two daughter cells. So this entire cycle, we call it as entire period of life period of a cell, we call it as cell cycle. So cell growth, it implies cytoplasmic increase is a continuous process. So cell growth itself is a continuous process. So you have to remember cell growth itself is a continuous process that is taking place. So DNA synthesis, it occurs only at a specific stage. So in the cell cycle, S stage or S phase is the particular division where the DNA synthesis occurs or DNA replication occurs at the S phase of the cell cycle. So the DNA synthesis occurs only at a specific stage. So here you can see the slides which are exhibiting the DNA replication. Okay, what is the type of replication and other things? Next year, you'll be studying in the molecular basis of inheritance. So usually it is going to be semi-conservative mode of replication, wherein you notice that they are going to have identical copies of DNA produced. So the newly produced DNA will be having 50% of the parental strand, and the newly synthesized strand in this daughter DNA is the daughter strand. Okay, so it is semi-conservative mode of replication because the newly synthesized DNA will be having 50% of the parental strand retained, and they are going to be identical copies to that of the parental DNA. So the daughter DNAs that are formed will be identical copies of the parent DNA because 50% of the parental strand is retained. That is why we call this method of replication as semi-conservative mode of replication. So this semi-conservative mode of replication and mechanism we'll be studying in the second year or your 12th standard when we study about the molecular basis of inheritance. Now you can remember DNA synthesis is by semi-conservative mode of replication. So this diagram indicates that type of replication. And here you can notice the cell is dividing into two by the direct enlargement of the nucleus, forming a dumbbell shaped structure. And then the nuclear lobes move towards the opposite poles, and there is a formation of two daughter cells. So this entire process is the mitotic cell division that you can remember about. So in this slide, you can notice the cell division stages, especially anaphase, you can notice the neighboring cells are exhibiting the prophase stage. So, but this is the Anaphase stage that you can notice in the cell division. A very beautiful slide for that exhibiting the Anaphase stage. Okay. So the duration of the cell cycle, it varies from organism and each cell type. So from organism to organism, the duration of the cell cycle varies, and also from the type of cell. So the duration of a typical eukaryotic cell cycle, that is example, human cell is about 24 hours. So the duration of a typical eukaryotic cell cycle, that is the human cell, is about 24 hours. So that you have to remember. In yeast, it is only about 90 minutes. So in yeast, it is only about 90 minutes that it takes for the cell cycle. But in case of human beings, it is 24-hour cycle. And I had already told you that in the cell cycle, so cell cycle, 23 hours is involved for preparatory phase or interphase, and only 1 hour is required for cell division. So the preparatory phase, it is a long duration time. It is not a resting stage, but the cell is getting prepared both cytoplasmically as well as from the nuclear point of view. They undergo replication and they increase in volume so that they are ready for the cell division to take place. So the preparatory phase in human beings is for nearly 23 hours, and the cell division takes only 1 hour. So the total cell cycle is 24 hours, of which 23 hours is for interphase stage and 1 hour is for cell division. Now, yeast is a unicellular fungi. Okay, so in this yeast, their entire cycle takes place within 90 minutes. That is why we have the statement that cell cycle duration varies in each organism and each type of cell. Okay, it might be different for a epithelial cell, muscular cell, the cell cycle might be different, even though it is in the same organism. And you can also notice that the cell cycle between different organisms, the duration also varies. In human beings, it is for 24 hours. In yeast, it is for 90 minutes. So not only it varies in the each and every organism, but also the cell type of that organism also might show differences in the cell cycle duration. So cell cycle has two basic phases, interphase and M phase. So the interphase and M phase are the two basic phases. So you can see this interphase is represented by this color, and M phase is almost this greenish color, isn't it? Yellowish green. So that is for short duration of time, whereas the interphase is long duration of time. Now, if you look into the interphase, what are the various stages that you notice? So they have this G1 or gap 1 phase, S phase or synthetic phase, G2 or gap 2 phase, followed by M phase, that is the mitotic phase. So interphase involves G1, S, and G2 phase. So G1 stands for gap 1 phase, S stands for synthetic phase, G2 stands for gap 2 phase. Okay, that is the interphase or the preparatory phase. M phase involves the karyokinesis and cytokinesis, that is the division of nucleus by the various cell division cycles, prophase, metaphase, anaphase, and telophase, and division of cytoplasm. We call it as cytokinesis. So finally, after the end of mitosis, you can find this cell cycle, you can find two daughter cells are being formed. So this is what you have to remember regarding the various stages of the cell cycle. Majorly, they have two basic phases, cell cycle, interphase and M phase. So interphase is also called as resting phase, but as I told you, rather we should be calling it as preparatory phase than resting phase. It is the phase between, interphase can be defined as the phase between two successive M phases. So it is the phase between two successive M phases. We call it as interphase. So the interphase includes cell growth and DNA synthesis. During interphase, two things are happening: the cell growth, that is cytoplasmic increase, and DNA synthesis. Both of them are happening in the interphase stage. And interphase lasts more than 95% of the duration of cell cycle. So you can just notice here, this entire cycle interphase, it is of the longest duration phase. Okay, more than 95% of the cell cycle duration of cell cycle is taken up by the interphase, and only the remaining 5% or lesser than 5% is the M phase. So the cell cycle lasts, the interphase lasts more than 95% of the total duration of cell cycle. Okay, so that you have to remember about. So now interphase has three phases. So I had already discussed about it. G1 phase is also called as gap 1 or antiphase. Okay, or you can also call it as post mitotic phase. Gap 1, G1 phase can also be called as gap 1 or antiphase or post mitotic phase. Okay, B is yes phase. The second one is the S phase, which we also call it as synthetic phase. And the third phase is G2 or gap 2 phase. So interphase has three phases: G1 or gap 1 phase, or interphase, antiphase we call it as, and this G1 can also be called as post mitotic stage. Then S phase is also called as synthetic phase. G2 or gap 2 phase it is called as, and it can be also called as pre-mitotic phase. Okay, so these are the different phases of interphase. See, in G1 phase or gap 1 or antiphase, majorly the cell is going to get prepared for the DNA replication. That will be, it's a preparatory phase for DNA replication. Second things, RNA and proteins are also being synthesized in this gap 1 phase or antiphase, or first G1 phase is nothing but gap 1 phase or antiphase or G1 phase. So it is the first growth phase. It is the interval between mitosis and synthesis. That is the DNA replication. So gap 1 phase, you can also define it as it is the interval between the mitotic phase and DNA replication. Okay, the main events, key steps that are involved in this gap 1 phase is there is a continuous growth of cell. So the cell grows continuously and cell becomes metabolically active. It prepares, major points are it prepares the machinery for DNA synthesis or DNA replication. And during gap 1 phase, there is synthesis of RNA and proteins. See, both gap 1 and gap 2 phase, they are involved in the synthesis of RNA and proteins. That you have to remember. So apart from that, the gap 1 phase also involves the preparation mechanism, prepares the machinery, DNA machinery for replication. So it's a preparatory for a phase for DNA machinery for replication. The DNA replication takes place in the synthesis phase, but the preparatory phase starts in the gap 1 itself. Okay, so there's a continuous growth of cell. So cell becomes metabolically active in the gap 1 phase. So these are the main events or key features that are involved in the cell cycle. The second one is synthetic phase, S phase or synthetic phase. In this phase, the DNA replication takes place by semi-conservative mode of replication. The DNA replication takes place. So the DNA doubles up. So the amount of DNA per cell, it doubles up, but the chromosome number remains the same. It is not going to change. So you can see that in this diagram. So before the DNA replication, they had only one sister chromatid per centromere. They had a single sister chromatid. But after DNA replication, the chromosome number has not changed. Only two chromosomes are there even after DNA replication. But you can notice that instead of a single sister chromatid, they have two sister chromatids formed because the DNA replication has taken place. So the number of chromosome does not increase, but the amount of DNA has increased after this DNA replication, which takes place in the synthesis phase. So, but chromosome number is not increased. In animal cells, the replication begins in the nucleus and the centriole duplicates in the cytoplasm. So there are two steps that are taking place in the animal cell. There is not only the DNA replication within the nucleus, but in the cytoplasm, there is the centriole also duplicates in the cytoplasm of this animal cell. So this centrioles are going to move towards the opposite poles, and later they are going to form the asters during the cell division stages. So there is duplication of centrioles also taking place in the animal cell. But in plant cell, there are no centrioles. So only in animal cell, you find that in the cytoplasm, apart from DNA replication in the cytoplasm, there is a duplication of centrioles, which will be forming the future asters with microtubules. Okay. The final phase of interphase is G2 phase or gap 2 phase. So G2 phase is also called as second growth phase. The cell growth continues. Synthesis of RNA and proteins continues. The cell is prepared for mitosis. So in the second gap or G2 phase, second growth phase it is, the cell growth continues. In this process, what you notice is the cell growth continues. The synthesis of RNA and proteins, which was taking place from the gap 1 phase, continues even in the gap 2 phase. And the cell is now prepared to enter into the mitotic cell division. It is prepared for mitosis. The next important phase is M phase. Okay, so in mitosis phase, what you notice is it represents the actual cell division. M phase represents the actual cell division or mitosis which is taking place. In human cycle, the mitosis lasts for only about an hour, whereas the interphase lasts for 23 hours. You should remember that. So the preparation takes longer duration, and it is the interphase is a preparatory phase rather than a resting phase. The M phase includes two steps: karyokinesis, that is nuclear division, followed by cytokinesis, that is the division of cytoplasm. So M phase includes two steps: karyokinesis, nuclear division. It takes place during prophase, metaphase, anaphase, and telophase. After the completion of telophase, the end of karyokinesis, so they enter into cytokinesis where there is a division of cytoplasm. So M phase includes karyokinesis and cytokinesis. Karyokinesis is the division of nucleus or nuclear division. Cytokinesis is the cytoplasmic division or division of cytoplasm. So some cells do not show any cell division at all. Example, heart cells or cardiac cells, they do not show any cell division at all. So many other cells divide only occasionally. So some of the cells don't divide frequently, they divide only occasionally to replace the damaged or dead cells. So the healing process of an injury or wound, as you can see, the healing process involves the cell division. So many other cells, they do not divide occasionally, they do not divide frequently, and only occasionally they divide to replace the damaged or dead cells, dead and worn out cells, or to replace the damaged cells, they undergo cell division. Okay. The cells which do not divide further, they exit the gap 1 phase. Okay, the cells which do not divide further, they exit the gap 1 phase and enter an inactive stage called as G0 or gap 0 phase. So this stage is called as quiescent stage. It is an inactive stage where they cannot undergo proliferation or they cannot multiply. So we call the stage as quiescent stage or gap 0 phase. So the cells that do not divide further, they exit gap 1 phase and they enter in an inactive stage called as quiescent stage. Okay, or it is also called as gap 0 phase. Such cells remain metabolically active, but they do not proliferate. That is, they do not get involved in cell division. So such cells remain metabolically active, but they do not proliferate or multiply. So the part two, first that completes our discussion of cell cycle. The second part is the discussion on mitosis. Okay, so mitosis It is the cell division occurring in somatic cells. So we also call it as equational division. The cell division occurring in somatic cells, we call it as mitosis. So somatic cells or vegetative cells, they are non-reproductive cells. You also know about it. So it is also called as equational cell division, as the number of chromosomes in the parent and the progeny cell, they remain the same. So mitosis is generally seen in diploid cells. Okay, so it is also seen in haploid cells of some lower plants and some social insects. So mitosis is generally seen in diploid cells, but it is also seen in haploid cells of some lower plants like gametophytic generation. So when they are undergoing the plant body in the gametophyte, so it is by mitotic cell division. Even though the gametophytic generation have haploid cells, they undergo mitotic cell division. That's an example of haploid cells of so lower plants and some social insects undergoing mitotic cell division. So the mitosis involves major reorganization of all the cell components. So the mitosis involves the major reorganization of all the cell components. So what important features of mitosis is it takes place in somatic cells or vegetative cells, or it is also called as non-reproductive cells. You can generally notice the mitosis. Mitosis is also called as equational cell division because there is no change in the ploidy level of the parent cell and the newly formed daughter cells or the progeny cells. So the number of chromosomes or ploidy level remains the same. So the parents are diploid, the newly formed progeny cells would also be diploid. So mitosis is noticed in diploid cells, but you can also notice mitosis in haploid cells of some lower plants and some social insects. Okay, like the female bees, they're all haploid. Okay, so there you can notice this sort of mitosis there. It involves major reorganization of all the cell components. Okay, so the mitosis involves the major reorganization of all the cell components. So here in this cycle, you can notice the somatic or vegetative cells, they undergo the prophase stage of it. So then they enter the chromosomes are much more condensed. The chromatin material gets highly condensed and chromosome material are visible. So then there is disintegration of nuclear membrane and nucleolus in the prophase stage, and the chromosomes are found freely floating in the cytoplasm. So when they enter the metaphase, all the chromosomes are arranged on the equatorial plane. So we also call this as metaphasic plate. And you can also notice that the asters, the centrioles are moved towards the opposite poles, and we call them as asters which have this microtubules. The centrioles with microtubules, we call it as asters. From this microtubules, the spindle fibers arises, which attach to the centromere of the chromosome. Now you can notice in both the uh, opposite side centrioles, the fibers are attached to the opposite ends of the centromere of a chromosome. All the chromosomes, they'll be having attachment of the spindle fibers from both the opposite poles. So when the spindle fibers condense during the anaphase stage, so when they get reduced, so it is almost similar to the tug of war. There is pulling from both the end. So what you can notice in the anaphase stage is the centromere splits longitudinally, and the sister chromatids also get separated. The centromere splits longitudinally, that is vertically there is a split of the centromere, and there is the separation of the sister chromatids, and they move towards the opposite poles by the condensation of spindle fibers during anaphase. So the chromosomes move towards the opposite poles, and you can also notice here there is only splitting up of centromere longitudinally. The number of chromosomes remain the same. So there is no change in the number of chromosomes in this anaphase phase. So the anaphase stage. Next is there is a reappearance of nuclear membrane and nucleolus. So the chromosomes are going to get uncoiled to form chromatin material. Then there is also reappearance of uh, or the reformation of endoplasmic reticulum and Golgi complex that is taking place in the telophase phase. So now this is the last stage of karyophynesis. During telophase stage itself, you can notice that there is a uh, furrow formed within the cytoplasm, and that furrow deepens and they meet at the center to form and they form a cross wall. So this separates this two daughter cells. The newly formed two daughter cells will have the same chromosome number or the same ploidy level as that of the parent cell. They have just increased in number. So we call this type of cell division, mitotic cell division, as equational cell division because there is no change in the chromosome number or the ploidy level. Okay, this is the entire process of mitosis in a nutshell. Okay, that is what we are going to discuss step by step in the coming slides. So the mitosis, the karyokinesis of mitosis has four stages: prophase, metaphase, anaphase, and telophase. Okay, so the prophase, metaphase, anaphase, and telophase, you can notice in the karyokinesis of mitosis. During the prophase stage, it is the longest phase in mitosis. Prophase is the longest phase in mitosis. It follows immediately after the S and G2 phases of interphase. So in the S and G2 phases, the DNA molecules are intertwined. So the DNA molecules are intertwined. So in prophase, you can find the condensation of the chromosomal material or chromatin fibers. So in key features of prophase are, so in the chromosomal materials, they are going to untangle and condense to form mitotic chromosomes. So now you can see distinct structures called as chromosomes. So the chromatin material is going to highly coil themselves, that is condensed to form a mitotic chromosome. So they are these mitotic chromosomes, if you look into, they have a centromere and they are composed of two chromatids which are attached to each other at the centromere. So you have two sister chromatids attached to each other at the centromere. That is what you notice in the chromosome. The centrosomes at the same time they move towards the opposite poles. In the animal cell, the centrosomes they move towards the opposite poles and opposite poles of the cell. Each centrosome radiates out microtubules, which we call it as asters. So the centrioles radiate out microtubules, which we call it as asters. And from this microtubules of this asters, the spindle fibers arise. The two asters together with so the two asters together with the spindle fibers, we call it as mitotic apparatus. Define mitotic apparatus. It implies the two asters with the spindle fibers together with spindle fibers. We call it as the mitotic apparatus. So that mitotic apparatus is forming. Also, you can notice that, so at the end of the prophase, they do not have Golgi complex endoplasmic reticulum, neither they have nucleolus nor nuclear envelope. So all of them are going to disintegrate. So the nuclear envelope, nucleolus, Golgi complex, endoplasmic reticulum, they disintegrate. At the end of prophase, they have disintegrated. Okay. In the metaphase, the nuclear envelope completely disintegrates. Hence, the chromosome spread through the cytoplasm of the cell. Initially, the chromosome is spread through the cytoplasm of the cell. The chromosome condensation is completed. So the chromatin material were getting highly coiled and condensed, isn't it? The chromosome material is condensation is completed. So I've already told you the best stage to study the structure of chromosome is at the metaphase stage. The best stage to study chromosome is at the metaphase stage. So now the chromosome condensation is completed, and they can also be easily studied under the microscope. You notice that the centromere will be having two sister chromatids. At this stage, you can notice it much more clearer. Okay, and the chromosome now they all come together to lie at the equator. So the plane of alignment of the chromosomes at metaphase, at the equator in metaphase, we call this as the metaphase place. So in metaphase, what you notice is the chromosomes come to lie at the equator. Initially, they were floating in the cytoplasm. Now they come to lie at the equator of the cell. So the plane of alignment of the chromosomes at the equator, we call it as the metaphase plate. Okay. The spindle fibers from both the poles are connected to the chromatids by the kinetocost. So the centromere has kinetocost, C-shaped commissural cups from which the microtubules arise. That is the kinetocost zone. So the spindle fibers are going to be attached to the centromere's kinetocost. From both the opposite poles, the spindle fibers are attached to the kinetocost found at two ends of centromere. Okay, so the spindle fibers from both the poles are connected by chromatids are connected to chromatids by their kinetocosts in the centromere. So that is what you can notice. And I have already told you when the condensation of the spindle fiber takes place, so they are going to the chromosome splits at the center longitudinally, and they are going to move towards the sister chromatids also get separated and they move towards the opposite poles. So during anaphase, the centromere will be having a single sister chromatid because of the splitting up of centromere. See, anaphase is the shortest phase in the mitosis. The longest phase in the mitosis is prophase. The shortest phase in the mitosis is anaphase. The centromere of each chromosome divides longitudinally, resulting in the formation of two daughter chromatids. Okay, so the sister chromatids are getting separated out because of the division of the centromere longitudinally, and the chromosomes of the future daughter nuclei, you can see that they are going to move towards the opposite poles. Okay, as the spindle fibers contract, the chromatids move from the equator to the opposite pole. So this is the late anaphase when you can notice that the chromatid fibers have contracted, the spindle fibers have contracted and the chromatids have moved towards the opposite poles from the equator. So this is the late anaphase stage that you can notice. So in anaphase stage, which is of shortest duration, there is longitudinal splitting up of centromere and the spindle fibers condense to move the separated chromatids. So each chromosome will be having a single sister chromatid. Okay. So in the telophase, the chromosomes cluster at opposite poles. So their chromosomes are going to cluster at the opposite poles and they are going to uncoil. The chromatin material is going to uncoil into chromatin fibers. So the chromosomes cluster at the opposite poles and they uncoil at chromatin fibers. They uncoil into chromatin fibers. The nuclear envelope they assemble around this chromatin fibers. So thus you can find two daughter nuclei are formed. So the nucleolus, Golgi complex and endoplasmic reticulum, they reappear during this phase. So the spindle fibers disappear. So these are the stages, key features that are happening in the telophase. So the chromosome clusters at the opposite poles and the chromosomes are going to uncoil to form the chromatin fibers. The nuclear envelope assembles around the chromatin fibers. So there are two daughter nuclei formed. And there is a reappearance of nucleolus, endoplasmic reticulum, and Golgi complex. The spindle fibers are going to disappear. So these are some of the key features of telophase. Now they enter into cytokinesis. After telophase, which is the final stage of karyokinesis, the cell now enters into cytokinesis where there is division of cytoplasm. Cytokinesis is the division of cytoplasm and they result in the formation of two daughter cells. You can notice there is a formation of two daughter cells here. So it starts when telophase is in progress. So the cytokinesis starts when the telophase is in progress. You can notice that already there is a furrow formed in the animal cell during the telophase stage itself. That furrow deepens up and they meet at the center to form a wall. So this furrow is going to deepen up, deepen, and they are going to meet at the center. So to form a wall. Okay, and that is how two daughter cells are separated. In case of plant cell, they have a cell wall. As a result of that, there is a formation of the vesicles. Okay, the formation of vesicles at the margins. So these vesicles are going to uh, grow towards both the ends, and as a result they are going to form a plate, cell plate like structure, which divides the uh, cells into two daughter cells they divide into, and this cell plate is later going to form the middle lamella. Okay, that is in case of plant cell. Plant cell has cell wall. That is the reason they undergo those sort of formation of the vesicles from the Golgi apparatus. So those vesicles are going to grow and extend outwards both the end, and they are going to form the cell plate. And this cell plate would have divided the cell into uh, two cells. The cytoplasm gets divided and they form two daughter cells. And this cell plate later would form in plant cells, later it forms the middle lamella. So whereas in animal cells, since they don't have a cell wall, so they are going to form a furrow, and this furrow deepens up to meet at the center to form the cross walls, and they are going to divide to form two daughter cells. So in animal cell, there is a cleavage furrow, which you notice from the telophase itself. So the cleavage furrow appears in the plasma membrane and it gradually deepens and joins at the center and divides the cytoplasm into two, and they are going to form the two daughter cells. This is happening in animal cell and it is a simple step. Reason is they don't have a cell wall. So in animal cell, the cleavage furrow is formed by the plasma membrane. It appears in the plasma membrane during telophase stage itself. This furrow, it gradually deepens and joins in the center from both the ends. They are going to deepen and join in the center, dividing the cytoplasm into two. There is a crosswall formed and they divide the cytoplasm into two, so which separates out to form two daughter cells. So there is a formation of cleavage furrow which gradually deepens and joins in the center, dividing the cytoplasm into two. So there are, the furrow is going to form a, when it deepens, it forms a plate, a cross wall. Okay, and they are going to divide to form two daughter cells. But in plant cell, the cytokinesis in animal cell is different. In the plant cell, it is different from the it is different in the plant cell when compared to animal cells because plant cells have cell wall. So in plant cells, the vesicles form Golgi bodies. Formed from the Golgi bodies, they are going to accumulate at the equator. So the vesicles are produced by the Golgi bodies, which accumulate at the equator. So it grows outwards. The vesicles grows outwards and meet the lateral walls. They fuse together to form the cell plate. So here they have formed the cell plate and it separates the two daughter cells. Later, the cell plate becomes the middle lamella. This is what is the cytokinesis in case of plant cell. Since they have a cell wall, their cytokinesis is different in plant cell when compared to that of animal cell. So the Golgi bodies secrete vesicles which arrange themselves in the equator. So the vesicles grows outwards towards the lateral wall and they fuse together to form the cell plate. The cell plate separates the two daughter cells. So later the cell plate becomes the middle lamella. So during cytokinesis, you can also notice that organelles like mitochondria and plastids gets distributed between the daughter cells. In some organisms, karyokinesis is not followed by cytokinesis, because of which, as a result of which, the cell becomes multinucleate condition or syncytium arises. So if there is no cross-wall formed immediately after the karyokinesis, the cell becomes multinucleate or syncytial condition. So it arises. Example is liquid endosperm in coconut. They'll be having the syncytial cells where there is no cross-wall formed. So the liquid of coconut, the free nuclear endosperm, we call it as. So the liquid endosperm in coconut is having this syncytial cells, cells with uh, large number of nucleus, because there is no cytokinesis immediately after karyokinesis. So this is the overall step or summary for the mitotic cell division. So the prophase, then in the metaphase, the arrangement of the chromosomes at the equator, which we call it as metaphase plate. So the splitting in the anaphase, there is condensation of spindle fibers, and it has similar to the tug of war effect on the chromosomes. So the centromere splits longitudinally, and the sister chromatids separate and they move towards the opposite pole. In the telophase, there is a reappearance of nuclear membrane, nucleolus, endoplasmic reticulum, and Golgi complex. The chromosomes, which are highly coiled, they uncoil and they are going to form the chromatin material. So there is a new reappearance of nuclear envelope encloses them. Okay, then there is the cytokinesis. So the telophase is the end of the karyokinesis, which is followed by the cytokinesis, that is division of cell cytoplasm, and the new daughter cells are formed, and both the daughter cells, newly formed daughter cells, will have the same chromosome number as that of the parent cell. So the ploidy level remains the same. So that is the reason mitosis is also called as equational cell division. So the significance of mitosis. So it produces diploid daughter cells with identical genome. So what do you mean by genome? I have already told you that genome is the total number of genes present in haploid set of chromosomes. So there is no change in the genome of the cell. So it produces diploid daughter cells with identical genomes to that of parent cell. So the sum total of genes remains the same even in the daughter cells. Okay, that is what it implies. Cells with identical genome. Why there is identical genome? Because there is semi-conservative mode of replication. During DNA replication itself, there is identical copies of DNA being produced. Why there is an identical copies of DNA being produced? Because 50% of the parental strand is retained, and the complementary or the daughter strand, which is formed, will have complementary to that. So the DNA copies that are produced during replication, which is semi-conservative mode of replication, is identical copies, because of which the daughter cells will also have identical genomes. Okay, so the mitosis helps to retain the same chromosome number in all the somatic cells. That is why we call it as equational cell division. So the mitotic cell division also helps in the growth of multicellular organisms. So the mitosis also helps in the growth of multicellular organisms. Mitosis in the meristematic tissue, they help in the continuous growth of plants throughout the life. So the mitosis in the meristematic tissue, they are found in the root tip and shoot tip. They are also found at the base of the internodes and at the base of the petiole. So we call it as intercalary meristem and lateral meristems. So the mitosis in the meristematic tissue, they help in the continuous growth of plant throughout their lifetime. Okay, so mitosis also restores the nucleocytoplasmic ratio, which gets disturbed during the interphase stage. So it restores the nucleocytoplasmic ratio. Mitosis restores the nucleocytoplasmic ratio that was disturbed during the cell growth. So mitosis also helps in the cell repair and replacement. So the dead and worn out cells are replaced. Cell repair and replacement. Example, cells of the upper layer of the epidermis, lining of the gut, and blood cells. They are repaired and replaced by the mitotic cell division. So the next type of cell division is the meiosis. I've already told you the backdrop of the slide shows an ovum, which is being approached by a male gamete. So the entire meiosis or reductional cell division takes place during the formation of gamete. So they are noticed during the gametogenesis. So gametes are generally haploid. So they have half the number of chromosomes as that of the parental cell. So for them to be haploid, there is a purpose. To maintain a constant ploidy level, they are haploid. So meiosis takes place in the germinal cells or reproductive cells. So meiosis results in the formation of four daughter cells and all of them are haploid. So since the number of chromosomes is half that of the parental cell, so we also call meiosis as reductional cell division. So meiosis takes place in germinal cells. Meiosis has two stages, meiosis 1 and meiosis 2, which forms immediately. So they are consecutively, they take up the stages. Meiosis 1 and meiosis 2 are successive stages. And prophase 1, there is genetic recombination, which takes place in the prophase 1 stage, which is very important stage of meiosis. Okay, so let us proceed with this meiosis or meiotic cell division. It is a division of diploid germ cells or the gamete mother cells, you can call it as like microspore mother cell or megaspore mother cell. So it is the division of diploid germ cells that reduces the chromosome number by half, forming haploid daughter cells or gametes. It occurs during gametogenesis. When does this meiosis take place? Especially during the gametogenesis. Gametogenesis means formation. Genesis means formation or development of gametes, formation of gamete. So next year you'll be studying in the reproduction chapters, spermatogenesis and oogenesis. Okay, so they are gametogenesis in human beings. We'll be studying that in the next academic year. So just remember about this stage. So the meiosis occurs during gametogenesis. So meiosis leads to haploid phase in the life cycle of sexually reproducing organism. So and the fertilization process, that is the fusion of male and female gamete or syngamy, what we call it as fertilization or syngamy of male and female gametes, and it results in the formation of the zygote. So fertilization or syngamy restores the diploid phase of the cell. So when both male and female gametes fuse, so haploid and haploid set of chromosomes, they are going to combine to form diploid phase in the zygote. So fertilization or syngamy restores the diploid phase uh, in the sexually reproducing organism. Okay, so the key features of meiosis are it involves two cycles, meiosis 1 and meiosis 2, but only a single cycle of DNA replication. So the cell cycle consists of a single cycle of DNA replication. In between meiosis 1 and meiosis 2, there is interkinesis stage, but that is not second interphase. It is just a, a transitional stage. So interkinesis is just a transitional stage. There is no synthesis of DNA in the interkinesis phase. So you should remember there is only one single cycle of DNA replication, but two cell divisions are taking place. So a single cycle of DNA replication is involved that is taking place in interphase before meiosis 1. So it involves two cycles. Meiosis involves two cycles, meiosis 1 and meiosis 2, but only a single cycle of DNA replication. Meiosis involves pairing up of homologous chromosomes and genetic recombination between them. So I told you that each and every cell of us or any organism, they have two sets of chromosomes. One set of chromosome is obtained from the male gamete, another set of chromosome is obtained from the female gamete. So the male chromosomes obtained from male gamete, we call it as paternal chromosomes, even though sometimes I call it as papa chromosomes, only for fun part of it. But when you are writing it, it is paternal chromosome. And another set of chromosomes is from mother side, mama chromosomes, which we call it as maternal chromosomes. Use the term paternal, maternal, not papa, mama. Okay, so paternal chromosomes, maternal chromosomes. Each and every cell of our body will have two sets of chromosomes, which we have obtained from our father's side as well as mother's side. And they are homologous, similar chromosomes like chromosome 1. There is a pair of chromosome 1 in us. There's a pair of chromosome 2 in us. So chromosome 1 derived from mother side, chromosome 1 derived from father side. So they are similar. So we call them as homologous chromosomes. It involves pairing of homologous chromosomes and genetic recombination between them. So genes are going to recombine between this homologous chromosomes. That is taking place during meiosis. So meiosis involves pairing up of homologous chromosomes, or we call it as synapsis, and genetic recombination between the homologous chromosomes. Okay, so meiosis 1 begins after replication of parental chromosomes to form identical sister chromatids at the S phase. So there is meiosis 1 is going to begin only after the S phase and G2 phase. So where there is replication of parental chromosomes to form identical sister chromatids at the S phase. Okay, so four haploids daughter cells are formed at the end of meiosis 2. Whereas in mitosis, it was only two daughter cells, whereas in meiosis, four haploid daughter cells are formed at the end of meiosis 2. Okay, so the stages of meiosis. So meiosis 1 and meiosis 2, two cycles are there. In meiosis 1 involves prophase 1, metaphase 1, anaphase 1, and telophase 1. Meiosis 2 involves prophase 2, metaphase 2, anaphase 2, and telophase 2. So the meiosis 2 involves prophase 2, metaphase 2, anaphase 2, and telophase 2. Meiosis 1 is where an important phase is prophase 1, which is an elongated phase which has five substages. So I've asked you to remember based on the behavior of chromosomes, there are five stages. So leptotin, zygotin, pachytin, diplotin, and diakinesis. So you can remember it sequentially by this mnemonics LZP D square, leptotin, zygotin, pachytin, diplotin, diakinesis. So the five phases based on the behavior of chromosomes. And what you notice in the metaphase one is the centromeres are not going to split longitudinally. Rather, the chromosome numbers gets reduced in the anaphase one because of the movement of the chromosomes towards the opposite poles. So there is no longitudinal splitting up of centromere taking place in anaphase 1. Rather, the chromosomes are arranged on the equatorial plate in such a way that the chromosomes move towards the opposite poles, and they are going to have only 50% of chromosomes are going to move towards the opposite poles. That is, haploidy gets established at the anaphase 1 stage itself. There is no splitting up of centromere longitudinally, neither there is no separation of the sister chromatids. Rather, in anaphase 1, you notice the separation of homologous chromosomes and the movement of this homologous chromosome towards the opposite poles. That is a unique thing taking place. Meiosis 2 is also called as haploid mitosis. All the stages are similar to mitosis. So meiosis 2, we also call it as haploid mitosis. And in metaphase 2, you find the arrangement of the chromosomes on the equatorial plate or metaphase plate. And in the anaphase 2, you notice that the centromere splits longitudinally and the sister chromatids separate and they move towards the opposite poles. Okay. So the haploids remain haploid because of this. Anaphase 2, where the centromere splits longitudinally and there is a separation of sister chromatids which moves towards the opposite pole. So these are some of the summary of meiosis. Okay, let us study in detail about them. So prophase 1, for 5 marks, many times they have asked this. It is typically longer phase and more complex. It includes 5 phases. Prophase 1 includes 5 phases based on the behavior of chromosomes. Those 5 phases are leptothin, zygotin, pachytin, diplotin, diakinesis, LZP, D square. So here they have given a summary of what takes place in these 5 phases. In the first phase, so the duplicated chromosomes start to condense. So the duplicated chromosomes, they are going to start to condense. Okay. So then in the zygote, the pairing up of homologous chromosomes takes place. The bivalent formation, that is homologous chromosomes pair up. So there is the pairing up of chromosomes, homologous chromosomes, and synaptonemal complex is formed, which holds this homologous chromosomes. So the pairing up of homologous chromosomes and formation of synaptonemal complex, which holds them together. So synapses begins in the zygote stage. So pachytene stage, what you can notice is synapses is completed in the pachytene stage and there is crossing over begins. So the non-sister chromatids of homologous chromosomes, see within the same chromosome, the chromatids, we call it as sister chromatids. So chromatids of two different chromosomes of homologous, we call it as non-sister chromatids. So you can notice the blue colored one and the red colored one, they are going to cross over. So there is crossing over of the non-sister chromatids. It is taking place in the pachytene. So completion of synapses and crossing over of the non-sister chromatids commences or begins in the pachytene. In diplotin, the synaptonemal complex, they disappear and there is a formation of chiasma. Since the synaptonemes disappear, the pairing, they were held together, now they separate, and as a result, you can see the crossing over place. There is a X-shaped arrangement, which we call it as the point of crossing over. We call it as chiasma. So the chiasma is visible in the diplotin stage. So the disappearance of synaptonemal complex and chiasma becomes visible during the diplotin stage. So the diakinesis, there is terminalization of chiasma and genes are going to rearrange. So the bivalent chromosomes, they are ready for metaphase. So this terminalization of chiasma, you can notice that, and there is the separation, there is genetic recombination having already completed in the diakinesis. Okay, so remember these are the various stages and synopsis of what is taking place in this prophase 1. Leptotin is also called as leptonema. The chromatin fibers, see they have just come from interphase. So prophase 1, you notice that the chromatin fiber becomes long, slender chromosomes. The nucleus enlarges. So these are the key features in leptotin. The chromatin fibers becomes long, slender chromosomes. They form chromosomes because of condensation of chromatin fibers, and they form long, slender chromosomes. The chromatin fibers becomes long, slender chromosomes. Nucleus size increases, nucleus enlarges. In the zygotine or zygonema stage, chromosomes become more condensed. Similar chromosomes starts pairing together. Synapses, the pairing together, zodiac, the pairing together or synapses takes place with the help of a complex structure called as synaptonemal complex. So the synaptonemal complex, it brings about pairing together of similar chromosomes by a complex structure called as synaptonemal complex. The pair chromosomes are called homologous chromosomes. Each pair of homologous chromosomes we call it as bivalent. Each pair of this homologous chromosomes, we call it as bivalent. So that pairing up of chromosome, formation of synaptonemal complex, and these paired chromosomes being called as homologous chromosomes. And this each pair of homologous chromosomes, we call it as bivalent. All these stages you can notice in the zygotine or zygonema stage. Okay. Then in the pachytene or pachinema stage, they are comparatively longer phase. Pachytene stage. So the pachinema, we also call it as the bivalent chromosomes, they split into similar chromatids. So this stage is called tetrads. So the bivalent chromosomes, they split into similar chromatids. Okay, they split into similar chromatids. So we call this as, this stage is called as tetrads. Okay, they split into similar chromatids. The appearance of recombination nodules at which crossing over occurs between the non-sister chromatids. So the recombination nodules are formed during pachytene at which crossing over occurs. It leads to recombination of genetic material on the homologous chromosomes. So from paternal, a fragment gets attached to maternal, maternal fragments gets attached to paternal. So this crossing over happens at the recombination nodules. Okay, and it leads to exchange of genetic material on the homologous chromosomes. So the crossing over is always between the non-sister chromatids. Recombination is completed by the end of pachytene. Genetic recombination is completed by the end of pachytene.
Sir,
So that is what you have to remember about. Yes.
sir, can you please explain patch 18 once again?
See, pachytene is pairing up of, there is a, let me get back to that slide. So it is a comparatively longer phase. The bivalent chromosomes, that is the similar pair of homologous chromosomes, the pair of homologous chromosomes. So the chromosomes split into chromatids. So you can clearly see the sister chromatids, two sister chromatids in each chromosome, you can notice. So if there is a pair of homologous chromosomes, they would be having four chromatids, two sister chromatids each chromosome you'll have. So this stage is called as tetrad. The bivalent chromosome split into similar chromatids. This stage is called as tetrads. So paternal chromosome one chromatid and maternal chromosome another chromatid. These two chromatids are non-sister chromatids. This non-sister chromatids from paternal and maternal, they are going to cross over. It is not that maternal chromatids, they are not going to cross over each other. It is always between non-sister chromatids. Sister chromatids are within the same chromosome. Non-sister chromatids are from two different chromosomes, that is paternal and maternal chromosome in a homologous pair. So non-sister chromatids are going to cross over at the recombination nodules form, and then crossing over takes place. So there is a tetrad formation in this pachytene. So wherein you find that the splitting up of chromosomes and the visibility of sister chromatids, you can notice that the chromatids separate, and this stage we call it as tetrad. Then appearance of recombination nodules, at which place the non-sister chromatids, they are going to cross over. And this leads to the exchange of genetic material from paternal to maternal and maternal to paternal. So exchange of or recombination of genetic material on the homologous chromosomes takes place. You can see this slide. So the maternal chromosome gets attached to paternal, paternal gets attached to maternal. So recombination is completed by the end of pachytene stage. Any other doubts you have?
No, sir.
Okay, that is pachytene. Then the next stage is diplotene. Diplotene, the synaptonemal complex, they dissolve. And the point at which crossing over takes place, the X-shaped structure, chiasma is visible. It is not that it was not there earlier, but now it becomes more visible because of the dissolution of synaptonemal complex. So the dissolution or disappearance of the synaptonemal complex occurs. The recombined homologous chromosomes of the bivalents, they separate except at the sites where they are crossing over. They separate at all the regions. These X-shaped structures where they have crossed over, we call that point as chiasmata. So chiasma is visible in the diplotin stage. In some of the oocytes, egg cells of vertebrates, diplotin lasts for months or years together. Okay, so for a few months or few years. So that is noticed in case of some of the egg cells of vertebrate. So what you notice is synaptonormal complex dissolution, the visibility of chiasma, so where the homologous chromosome, the bivalents separate at all the points except at the site of crossover. So this X-shaped structure is called as chiasmata. So diakinesis is the final stage of the prophase one. So terminalization of chiasmata. The chiasmata separates the X-shaped crossing over, they separate. When they are separating, you can notice that the blue colored chromosome gets a fragment of the red one, and the red one gets a fragment of blue color. So there is, you can notice that the genetic recombination has taken place. So terminalization of chiasmata, chromosomes are fully condensed, and the meiotic spindle fibers originate from the poles to prepare the homologous chromosome for separation. Nucleolus and nuclear envelope disappear. So you can notice that the disintegration of nucleolus and nuclear envelope, formation of spindle fibers, these are all getting ready for the metaphase phase. See, diakinesis is a prophase stage. So the disappearance of nuclear membrane, nucleolus, endoplasmic reticulum, Golgi complex, all that takes place, okay, in the at the end of diakinesis. Then there is the appearance of spindle fibers. The next stage is metaphase 1. How is metaphase 1 different is, here the metaphase 1, the arrangement of chromosomes you should see. So the spindle formation is completed in the metaphase 1. The chromosomes align on the equatorial plate as per their homologous chromosomes, they get arranged. They arrange at the equatorial plate. So the microtubules from the spindle attach to the pair of homologous chromosomes. Not for the, see, the chromosome are not attached by the opposite spindle fibers, opposite pole spindle fibers. They're attached at only one end. They are attached by the to the pair of homologous chromosomes. Okay, so from the spindle, the microtubules from the spindle, they attach to the pair of homologous chromosomes. Not at both the ends of the centromere, only one end of the centromere. So the metaphase arrangement of chromosome itself is in such a manner that the homologous chromosomes get separated during anaphase. So there won't be any splitting up of centromere. There won't be any separation of sister chromatids. That does not take place because of this type of arrangement of chromosomes in the metaphase 1. So the microtubules from the spindle attach to the pair of homologous chromosomes at only one end. Okay. In the anaphase, what happens is the homologous chromosomes, they separate by the condensation of the spindle fibers. The homologous chromosomes separate while sister chromatids remain associated at their centromeres. So there is no division of centromere, then there is no separation of sister chromatids in anaphase 1. Only the homologous chromosomes are separated. So when the homologous chromosomes are separated, you can notice at each pole there would be 50% of chromosomes each. Okay, so as a result of it, haploidy has already started. So the four chromosomes are not there at each pole. Rather, you can notice two chromosomes. Okay, so the homologous chromosomes are separated in the anaphase 1, while sister chromatids and centromere, they remain as they are. So in telophase 1, the nuclear membrane and nucleolus reappear, and two haploid daughter nuclei are formed. So two haploid daughter nuclei are formed, and we call this as dyad. So reappearance of nuclear membrane, nucleolus, endoplasmic reticulum, Golgi complex. So we call that as dyad. So two daughter nuclei are there. After this, the cytokinesis may or may not occur. So this dyad stage is the reappearance of nuclear membrane and nucleolus within the parental cell, the telophase stage. Okay, we call that as dyad. That is before cytokinesis. After this dyad formation, the cytokinesis may or may not occur. So there is a short interphase after this telophase 1. So it is followed by meiosis 2. The short stage between two meiotic divisions, we call it as interkinesis. So there is no significant change in interkinesis. It is just a short phase before transition to meiosis 2. DNA replication does not occur at interkinesis phase. Please remember that there is no DNA replication at interkinesis. It only takes place before meiosis 1 in the interphase stage. So there is no DNA replication during this interkinesis phase. Okay. In the meiosis 2, I'll just another 2 or 3 minutes, I'll complete it. So it resembles the mitosis. So meiosis 2 is also called as haploid mitosis. It has four phases: prophase 2, metaphase 2, anaphase 2, telophase 2. So what you can notice here is in the prophase 2, there is disappearance of nuclear membrane and nucleolus of the both the daughter cells. Then the metaphase 2, you find that the chromosomes are arranged at the equatorial plane. So in the metaphase 2, so the chromosomes are arranged at the metaphase or uh, equator, or we call it as metaphase plate, and the spindle fibers are attached to both the ends of the centromere from the opposite poles. The opposite pole spindle fibers attached to both the ends of the centromere. So this is what you notice, which was not seen in metaphase 1, and this results in the anaphase 2, the centromere splits longitudinally and there is separation of sister chromatids. So the sister chromatids moves towards the opposite pole, and in telophase 2 in both the daughter cells there is a reappearance of nuclear membrane, nucleolus, endoplasmic reticulum, and Golgi complex. All of them reappear. Then they undergo cytokinesis. So cytokinesis results in the formation of four daughter cells, four haploid daughter cells. That is what you notice in case of meiosis 2. So the anaphase stage, you can notice the splitting up of centromere and sister chromatids. Okay, that is why meiosis 2 is also called as haploid mitosis. All the stages takes place, okay, which I have described about. Okay, so the sister splitting up of centromere and separation of sister chromatids. Telophase 2, reappearance of nuclear membrane or nuclear envelope, nucleolus, endoplasmic reticulum, Golgi complex, they're all going to reappear. And cytokinesis is followed, resulting in the formation of tetrad of cells. Dyad, we called it as in meiosis 1, now we call it as tetrad of cells, four haploid daughter cells. Okay, these are the complete stages. So significance of meiosis, it conserves the chromosome number of each species by the gametes having haploid. So when they undergo fertilization, the chromosome number remains the same as that of the parental cell when they undergo fertilization. So meiosis help in the conservation of chromosome number of each species. Meiosis causes genetic variation due to crossing over in the population of organism, and variation itself is a raw material for evolution. So for evolution to take place, this genetic recombination is very important. Okay, so that completes our summary of the entire topic. Okay, so we'll wind up with this session.